Hello and welcome to the GCSE Biology tutorial for topic B3 Infection and Response. This tutorial will cover communicable diseases, prevention and treatment. Starting off with, what are communicable diseases? Well, these are diseases that can be spread and passed on, so they're the opposite of non-communicable diseases. You need to know that pathogens are microbes that cause disease, and there's 4 types of pathogens. Bacteria, and these are about 100 times smaller than your body cells and reproduce very quickly. In fact, most bacteria can reproduce once every 20 minutes, as shown here. They produce toxins in your body that make you feel ill. Viruses are smaller than bacteria, about 100 times smaller, and they're not living, so they need a host to live off. They enter the cells and reproduce quickly, then burst out of the cell, causing infection. Protists, these are single-cell organisms and they're eukaryotes, so they do have a nucleus. They're parasites and they damage living cells, and they can be transferred by a vector, for example, malaria and mosquitoes, which we'll talk about later. Finally, fungi. Fungi can be single-celled or bigger, and they enter living tissue and cause disease. They've got a body made of little thread-like structures called hyphae, and then they produce spores, which causes them to reproduce. So, you do also need to know how, in particular, viruses replicate. When a virus enters a cell, it uses the cell's outer coat and starts to strip it of its protein. The nucleic acid in the centre of the virus is then released. The acid gets into the cell's manufacturing system, so that means it gets into the DNA. The cell then ignores its own chemical needs and starts to make the new viruses. So the virus is taking over the DNA of the cell. The virus then starts to multiply and the cell is destroyed, and then the cell bursts open and all the new viruses are released to then infect others. You do need to know how different communicable diseases are transmitted. The examples you need to know are here in this table. So, one way they could be transmitted is through contaminated water. The most common disease that's transmitted like this is cholera, and that's bacteria. The symptoms of cholera are severe diarrhea, dehydration, and vomiting. Transmission examples also include air. So 2 diseases that you need to know could be transmitted through the air are the flu, so influenza, or measles. Influenza and measles are both caused by viruses, and the symptoms you will need to know of each as well. So the flu causes fever and coughing and other flu symptoms. Measles causes a rash on your skin, a red rash, and it can lead to fever and more severe conditions like pneumonia and brain infection. Another mode of transmission is direct contact, touching. Not many diseases are transmitted like this, but there is some. An example you need to know is athlete's foot, which is a fungus. This is picked up from swimming pool floors and towels and makes the feet inside, in between the toes, itchy and the skin starts to flake away. Vectors, we talked about these earlier. This is when an organism carries the disease but is not actually infected from it themselves. For example, mosquitoes carry the protists for malaria, but they don't have malaria themselves. If you're bitten by a mosquito carrying malaria, you can then get infected. And malaria causes fever and it can be fatal. Protection from mosquitoes includes using sprays and using mosquito nets. Another mode of transmission is bodily fluids, so unprotected sex or sharing needles. This can cause the virus HIV to be spread. Initially, HIV causes flu-like symptoms, but eventually it will attack your immune system. It can be controlled for a long time by antiretroviral drugs, but eventually the immune system is destroyed and it leads to AIDS. These days, the life expectancy of someone with HIV is a lot longer because these drugs are being developed all the time. Another STI, or sexually transmitted infection, is gonorrhea, and this is a bacterial infection. And the symptoms include pain when urinating and a green-yellow discharge from the vagina or the penis. And these can be treated with antibiotics. One way to prevent them is to use condoms during sexual intercourse. Another mode of transmission is through food, especially uncooked meat. An example of this is salmonella, which is a bacterial infection. And this causes fever, cramps, vomiting, and diarrhea. So it's really important that you cook your food all the way through. Barbecues, for example, are places where sometimes salmonella can occur. You also need to know about the transmission of diseases in plants, which is in the next part of the tutorial. In general, how can transmission be prevented? Well, just simple hygienic measures like washing your hands, disinfecting areas in the house, mainly the kitchen, the bathroom, destroying the vector itself, for example, the mosquito, or destroying the habitats where the vectors live, and of course vaccination, which we'll talk about later. So, what does your body do to fight against disease? Well, there's two barriers that your body has before the pathogen gets into your blood. Physical barriers. This includes your skin, which is a natural thick barrier that stops pathogens getting in and also produces oils that kills pathogens. There's hair and mucus in your nose that traps pathogens, and your respiratory system, so your trachea and your bronchi, have cilia, which are tiny little hairs, and mucus, which traps the pathogens and then sweeps them out. There's also chemical barriers. So your stomach has hydrochloric acid in it that kills pathogens. So a lot of the food that you eat, you won't get ill from because the bacteria is killed by the hydrochloric acid. Your tears also contain an enzyme that kills pathogens. Now, if those barriers don't work and pathogens do get into the body and they get into the blood, then you have a next stage of your defense system. And this involves your white blood cells. White blood cells work in three ways. They either engulf the pathogen, so don't say they eat it, they go up to the pathogen and they surround themselves around it and destroy it, so they engulf it. And these white blood cells are called phagocytes. Another thing that white blood cells can do is they can produce antitoxins, and those are against or anti the toxins made from the bacteria. And the third way is the production of antibodies, and this is done by white blood cells called lymphocytes, in particular B lymphocytes. So pathogens have unique antigens on their surface, and if they get into your body or into your blood, then B lymphocytes make specific antibodies that lock onto the specific antigen and then destroy it. This then triggers more antibodies to be made quickly. These antibodies stay and remain in your blood, and if you get infected from the same pathogen again, you won't become ill because you already have the antibodies in your blood. So you're said to now be immune, and that's one of the reasons that in most cases you wouldn't suffer from the same disease twice. Now, to prevent the spread of diseases, you could get a vaccination. So what actually is a vaccination? Well, it's an injection of a dead or weak inactive version of the pathogen. So they're actually giving you a little bit of the pathogen, but a weak version of it, or a dead version. Once you are injected with this, then you start to make specific antibodies. So your lymphocytes make specific antibodies recognizing the antigen on the pathogen. Now, they then remain in your blood, and if your body is then attacked by the live pathogen, so if you then actually do get the disease into your blood, the antibodies quickly reproduce and kill it straight away so you don't get ill. So a vaccination doesn't stop the pathogen getting into your body, but it stops you getting ill from it because you kill it straight away. If you look at this graph here, it shows that after you've got the same disease twice, so the second time you get it, your response is a lot quicker because you've already got some of those antibodies and they can reproduce very, very quickly. So it's a little bit like how the vaccination works, because you've already been given some of the weak version of the pathogen, so you have some antibodies in your blood already, and the second time, if you do actually get it, you'll kill it off straight away. An example of vaccination is MMR, which is the measles, mumps and rubella vaccination. Now, of course, there's lots of advantages of vaccinations. They can control these diseases and really prevent the spread. In some cases, they can eradicate and totally wipe out a disease like smallpox, which no longer exists. And they can prevent an epidemic, which means a global disease taking over. Especially if lots of people get vaccinated, it means that less people will then have a chance of getting it. However, there is some concerns. There can be very rarely some side effects, and it doesn't always work. It doesn't always cause immunity. But again, this is rare. These graphs just show some information regarding this. So this graph here shows the number of cases of measles, and then after the vaccine has been introduced, you can see that the number of cases went down, but so did the number of people who started to get the vaccine. And then there's been campaigns to make sure people get the vaccine as well. This chart here just shows how herd immunity works. If no one is vaccinated, then a contagious disease can easily spread. If some people are vaccinated, then the disease can still spread quite quickly. If most people are vaccinated, then the chance of the disease spreading is very minimal, as you can see here. And that's called herd immunity. So not everyone is vaccinated, but the majority of the population. So here are some key questions. If you pause the tutorial now and work through, give yourself 10 minutes. Okay, how did you get on? 3 ways in which communicable diseases can be transmitted. So any of these 3, the air, water, food, bodily fluids, vectors and contact. The 4 types of pathogen are bacteria, viruses, protists and fungi. How do antibodies defend against disease? Well, they are produced from white blood cells, or B lymphocytes, and they target specific antigens on the surface of the pathogen and attack and destroy them. What are the advantages of vaccinations? Well, they control the transmission of the communicable disease, they can wipe out the disease completely, and they can prevent epidemics. You also need to know about different drugs. So there are drugs like antibiotics. You need a prescription to get these in this country, and they kill bacteria, so they don't kill viruses. A common example is penicillin. There's also painkillers. They relieve the pain, but they don't actually kill the pathogen. So for example, aspirin or paracetamol, they'll lower your temperature, for example, but they're not going to kill or destroy the pathogen. Sometimes bacteria can become resistant to antibiotics. Most common example of this is MRSA, and that's a bacteria that actually increased in hospitals. It became resistant to a particular antibiotic, and that strain that became resistant then reproduced. And when it reproduced, it passed on that resistance. So more and more of the bacteria became resistant to the antibiotic. This is an example of natural selection, which we talk about in another tutorial. So it's important that doctors don't just give prescriptions for antibiotics for anything, but only things that are severe enough to need them. And also, even if you're feeling better, you must finish the full course of antibiotics. So, how are drugs developed? Well, a lot of drugs actually started originally from plants. For example, aspirin, which is a painkiller, is actually found naturally in willow. And digitalis treats heart conditions, and that's found naturally in foxgloves. But in any testing of any drug, that takes 4 main stages. First of all, preclinical tests on human cells and tissues. So in the lab, they'll test on cells and tissues and see how the drug impacts this. But this isn't the same as an actual system, a body system, an organ system. So the next stage does require tests on live animals, normally 2 animals to test the drug thoroughly. And this is to find the effect and find the best dosage. Some people are against testing on animals, others believe that it is needed before it's given to humans. The third stage would be clinical trials on human volunteers. And this is to check for any harmful side effects. Obviously, severe side effects would have already been eliminated. And also to find the optimum dosage, so really fine-tune that dosage. And what this involves is using healthy volunteers and giving some people the drug and giving some people something called a placebo, which is not the drug, but not telling them. Because sometimes when people believe they're being given a drug, it makes them feel better psychologically. So the placebo effect is almost tricking people. So you know that if they are feeling better, it is genuinely because of the drug and not just because they think they should be getting better. Before the drug is manufactured, it is also reviewed. There's also peer reviews that happen, which means that other establishments, other industries will test the drug as well before it is manufactured. The development of a drug can take up to 10 years before it is released. You will need to know in particular about something called monoclonal antibodies. We already know what antibodies are. Monoclonal, those two words are important. They're basically identical antibodies, clones, and they're produced by the B lymphocytes, which we talked about before as a type of white blood cell. How are they made? Well, they're made by cloning white blood cells, so all the antibodies are identical, and that means they can target just one specific antigen. And how you make them is you would insert the lymphocytes into a tumour cell, because tumour cells grow rapidly and easily, and that means you can clone whatever you insert into them. So a mouse B lymphocyte and a tumour cell can be fused together to make something called a hybridoma. And then the hybridoma is cloned and to produce identical antibodies. You can see this in this diagram here. So this mouse is vaccinated and the mouse will start to make antibodies. And then the spleen cells that form antibodies are collected and they're fused with the tumour cells. And then this forms a hybridoma and then that's cloned and then you make the antibodies. The good thing about monoclonal antibodies, they only target one type of antigen, so they can be used to target specific cells in the body. Here's some uses of them. One, pregnancy tests. So in a pregnancy test, there is monoclonal antibodies that target a hormone called HCG that you would only make if you were pregnant. Pregnancy tests have the monoclonal antibodies stuck to blue beads on a test strip. If you then wee or urinate on the strip, the monoclonal antibody in blue beads bind to the hormone in the urine. This then moves the blue beads up the strip and then turns it blue. Blue indicating that you will be pregnant. If you're not pregnant, then there will be no HCG hormone and so it wouldn't bind to the blue beads and so it wouldn't turn blue. Monoclonal antibodies can also be used to treat cancer because they can target specific cells. So if you know the marker on the outside of the cancer cell, which is called a tumour marker, then you can make monoclonal antibodies to target just them and they'll bind to it and kill it. And that means that only cancer cells are destroyed and not healthy cells. So some of the issues with chemotherapy and radiotherapy is it can also kill healthy cells. This kind of treatment would only kill the cancer cells. You can also use monoclonal antibodies for scientific research. So to find out more about the levels of hormones in the blood or locate specific cells, and you could use dyes to try and find where these cells are. The advantages of monoclonal antibodies are they kill specific cells and they don't damage the healthy ones. There is concerns with side effects. At first they didn't think there would be some side effects from monoclonal antibodies, but there has been some side effects with them, so more research needs to be done with them. There's a practical for this topic where you need to know about the effect of antibiotics on bacterial growth. For this, you would need to use an agar plate. Remember, an agar plate is a culture medium where nutrients are provided for bacteria to grow in. So it's like a hard jelly that bacteria like to grow in. You get little paper discs and soak them in different concentration of antibiotics and then place them on the agar plate. You then would cover the agar plate with a specific bacteria. And you'd also need a control. A control is a disc that has not been put in antibiotic and just put in sterile water. And then you'd leave this in an incubator at 25 degrees for 2 days. Afterwards, you'd look at the dish and you would look at something called the inhibition zone. The inhibition zone is the area that's clear here. And the clear area indicates that bacteria is being killed by the antibiotic. This is antibiotic here, there's another antibiotic here and here. This bigger one here indicates that this is the better antibiotic because it's got a bigger inhibition zone, so it's killed more bacteria. You can also calculate the area of the inhibition zone by using area equals pi r squared. Remember, you need to look at the diameter of the circle as well. Now remember, any practical involving culturing bacteria, you need to make sure that you use a sterile wire for inoculating. So you transferring bacteria, use a sterile wire loop, and you sterilize that by running it through a Bunsen flame. You would also avoid contamination by sterilizing the Petri dish, by storing the dish upside down, and also not taking the lid off and sticking it around the side loosely to stop anything growing. Once you've finished with the agar dish, you would then destroy it in an autoclave. So, here's a 6-mark question. If you take 10 minutes, pause and try and answer this question. Explain how vaccinations can prevent a person from catching rubella. Suggest why vaccinating a large amount of the population reduces the risk of the disease spreading. Okay, so you need to split this answer into 2 parts. First of all, what is a vaccination? So that involves injecting a dead or inactive version of the rubella virus into the body. Okay, if you remember, the disease they're talking about is rubella. It doesn't really matter what the disease is, it's the same principle. The virus that you've then injected has specific antigens which cause the body to make antibodies to attack them. Now remember, it's just a weak version of it, so you're not actually going to get rubella. But those antibodies remain in the blood. Then if the person does get rubella, the antibodies are reproduced quickly and defeat the virus before it can make the person ill. And that means that person will not then get and suffer from rubella. Now, if a large percentage of the population is vaccinated against rubella, then less people can pass it on. That means in turn, even people who've not been vaccinated have got a less chance of catching it because less people will have it. And that's called herd immunity. The important words to mention in this answer would be antigens and antibodies, because that shows that scientific knowledge of knowing that antibodies are what your white blood cells make and antigens are on the surface of the pathogen. Remember, the pathogen is the virus or the bacteria or the protist or the fungus or whatever the microbe is that's causing the disease. So, in summary, we've talked about communicable diseases, prevention, fighting diseases, vaccinations, drug development, and monoclonal antibodies. And the practical was looking at the effect of antibiotics on bacterial growth. If you look at the next tutorial for the second part, that will conclude this topic. Thank you.